Members, ladies and gentlemen. Today, on the occasion of the evening webinar on the topic of BNT, Bio Nanotube of Escherichia Coli K-12, its engineering and healthcare system, organized by the Chemical Engineering Division of West Bengal State Center. Let me take this opportunity to extend a warm welcome to you all and our panel speaker. Nanotechnology is an area of research and manufacturing technology of global interest, which deals with a variety of materials produced at a nanometer scale. Several attempts of transition deoxyribonucleic acid in biology and nanotechnology from ideal test tube conditions of to living systems have been carried out in recent years. BNT, biotechnology, bio nanotube of Escherichia coli K-12, is the recent breakthrough concept of the chemical engineering. The scientific applied engineering concept of BNT is therefore considered unique because it covers the understanding of nano level of molecular transport of chemical engineering, the molecular biology, and microbial infection. Its colonization mechanism, compatible immobilization techniques, and the new approach of mathematics through which the said BNT can in future replace the CNT carbon nanotube and to protect patient CNT immobilized nanomedicine and their side effects. The study of BNT in different chemical engineering transport concepts, microfluids for drug delivery, for development of recoveries of cancer patients is now becoming important at present in the field of medical and chemical science and engineering environment, where the transport of molecules has predominated. Escherichia coli or E. coli, which is known as, is a bacteria that is commonly found in the lower intestine of warm-blooded organisms. In today's webinar, hopefully, it will be addressed the engineering synthetic DNA nanostructure and develop methods to increase their lifetime in an all Escherichia coli cell-free transcription translation system. Today, we have our panel speaker, Professor Dr. Brahmo, who is a distinguished personality in the field of chemical engineering and one of the distinguished pillars in the field. Let us join together and explore much on the topic. I hope you will enjoy today's deliberation in a graceful manner. Thanks and welcome you again.
Thank you, Dr. Das, for your nice welcome address. May I now request Professor Dr. Shilpa Tripathi, Chairman, Chemical Engineering Division Board, to kindly address the occasion. Mr. Shen, Dr. Tripathi, is he present? Has he joined? Mr. Shen, can you confirm?
I believe she's not there.
May I now request Chairman Kate, Dr. G. Ranganath, to kindly deliver his address. Mr. Sen, can you confirm whether Dr. Ranganath is present?
Hello, Mr. Datta. I think Nobody is there. I can see the panelist. Dr. G. Ranganath is not there.
Okay, so I believe President and Vice President, they have also not joined.
No. You can just carry forward the program with the speaker directly.
Okay, so let me request our panel speaker, Professor Nitosh Kumar Brahmo, who is also the chairman of Chemical Engineering Division of West Bengal State Center and as well as immediate past honorary secretary of West Bengal State Center, to kindly deliver his address. Over to you, Professor Brahmo.
Professor Brahmo, please start.
Hello, can you hear me?
Yes, it is audible.
Yeah. Now I'm just transferring my slides. Just a minute. Desktop আছে। Desktop. এই যে। Yeah. So can you see my slides?
Yes, it is visible.
Yeah, it's visible. Okay, thank you very much for your kind permissions and taking some time to transfer in the webinar section. And now I start. The BNT, Bio Nanotube of Escherichia Coli K-12, is engineering and healthcare system. Can you see that? No? Okay. The next slides. Next slide, we have it over. In the abstract, I have described the nanotechnology has potentially revolutionized the global food, agriculture, medicine, material, metal, and cement processing. It is true. Nanotechnology has also revolutionized the coating and welding system of the metal industries. And in present time, to move towards the green nanotechnology without side effect, because the most of the case, whenever you are talking about the nanotechnology applicable to the medicine, it has given a lot of side effect because of the graphite as well as the carbon molecules, which is remaining in the carbon nanotube CNT. The present use of CNT, carbon nanotube, in chemotherapy provide many side effects and kills the patient due to side effects. To minimize the side effects, especially it's in healthcare, the author discovered the BNT of genetically engineered Escherichia coli K-12, which is universally and recipient bacterial strain, which remains actually Escherichia coli. The Dr. Escherichia coli discovered these bacteria is actually remaining in our intestine, and it has been modified in Yale University by mutational process as the best recipient and one of the best engineering activities involved, the bacteria, which is already well known to everyone, even in the school and college universities, college students, university students in the research areas. Is a fantastic tool after discoveries of the double helix, the DNA double helix by Watson and Crick. The people started to ask which will be actually the best recipient to work with that double helix and to discover its potential activities to the hybrid model, not the PO model. You understand that whenever you say hybrid, it is a mixed DNA technology involved in the bacteria. So there are a lot of wild type and the donor and the hybrid strains of existence in the laboratory of the world, which are being used mostly from different ways to understand is the vast potential activities of the microbes. Louis Pasteur, one of the examples, who told you cannot underestimate the power of microbes, and that has proved by Escherichia coli as well as the DNA double helix how fantastically they can design the new models of molecular activities of the cells. To impose the chemotherapy and cancer patient without side effects. And the most of the CNT, carbon nanotube, which has been used in the drug delivery because of its structure, I'll be coming to the slides, because of its structure, they can be immobilized in the carbon nanotube and they can be transported through blood transfusion to the cancer patient and targeted to the cancer cell. It is well known to everyone that the CNT has been used for the chemotherapy and immobilizations of the cancer drugs in the CNT. The methods of what is it? From methods of the print cell preservation, the methods of food and cell preservations are also being imposed by applications of nanotechnology. Copper, gold, silver, and their combinations of electrons, ionic configuration improved the systems of the preservation, metal, and finally food and medical aspects of medicine and drug nature. Due to increasing surface area of the nanomolecules, particles, the scientists could potentially use to deliver and sustainable natures of the environment. The main problems of our environment, as you know, the sustainability. Most of the industries are very much aware now how to recycle everything to the zero. And one of the best way that they are thinking that they will be processing the metal, we will be processing the medicine, we will be processing any molecules of the biological aspect, that it should not give any side effect, means it should not remain unused in the environment. And this process brings us also the sustainable concept of the continuation. That means our main target to sustain our environment without any destructive attitudes. One of the best example is the deplantation. Best example is the fire. Best example is how they destroy within a few hours, the total greeneries and the environmental surviving animals. It's a very dangerous situation that we are living. And that's why we are also very much aware about the side effects. Means I will not make any technology, I will not make any environment that will kill the innocence of the environment living creatures. This has also been proved in the evolution that how the animal fights, dinosaurs and what you can say, rhinoceros, dinosaurs, and the flying birds like reptiles, Archaeopteryx, they have struggling from survivals, and so Darwin said also survival of the fittest. That means we should then survive if we be fittest to the environment. So to make not to be disturbed and not to be changed the environment, we are therefore very much aware that we will create no side effect instrument, side effect metal, side effect chemicals that will be used in the human society. The definition of the tube and bio-nanotube could be explained in such a manner in the electron microscopes that you can see. You see the one microns, how will be 1 millimeters, the length, how the tubes can be observed, and which has already been described by the process that these tubes, why so important, as already we have discussed about the CNT, that how the CNT is being used, but their side effects, we are not recovering, sorry, we are not trying to manage, and sure, the scientists are struggling to modify the CNT by different metal combination, by different carbon combinations, etc., etc. But the thing is that we have not thought, or we are not thinking also at present days too, that how the microbes can also be involved in such type of transport, in such types of immobilization, in such type of defense. For example, that the author who is speaking, he has also thought how the antibiotic could be avoided from the world to increase the sustainability of the patient and so-called infected people by the means of anti-adherent immune response, AAIR. So this is one of the most important issues, how AAIR, anti-adherent immune response, could be sustainable if we can really realize and understand that these microbes can be involved to protect their wild type infections, their infections by the means of genetically engineered hybrid strain with the help of the K-12, the Escherichia coli, which can be modified by plasmid transformation. That means that strain is compatible to our body, has already been described, and it can take only that portions of information which are actually involved to damage the patient as well as to kill the patient. And if we could manage this part into Escherichia coli K-12, then we can make one hybrid strain which can provide this type of anti-adherent immune response. Only that portions can be protected. That means the best example we can say in simple, that when our bacteria going inside, the infective bacteria, which makes the diarrhea, which makes the fever, which makes also different types of infection, And recently the corona, I can tell you that's also microbes, but it is more smaller than the bacteria. It is a nano level structure. Bacteria is a micro level structure. That is also being imposed, also called being manipulated, could also be manipulated by the help of anti-adherent immune response. Mean, if we could isolate that type of gene, which is a DNA, which is actually infective, which is actually making the patient virulent, also called the killing the patient, means by adhering, by making the infection larger, whatever it might be. It could be a diarrheal form, it could be enterobacteria groups of coughing, and also other respiratory system, and whatever it might be. And if we could develop some immune responsive activity, then this can be utilized also in the corona system, as well as also in the infected system of any bacterial process. The origin of the tube and flow concept. Now, man can say that, well, adherence, PMDA, everything is been shown in electron microscope, is shown in the process of isolation and mice experiment, etc. But how could you say that these PMDA can actually be utilized as a nanotube and can be utilized in chemotherapeutic system in the physiology, and can carry some fluids and can support the flow of fluids, etc., etc. That should be proved. So it's accepted that the concept is very, very interesting. The anti-adherent immune response was identified by the author in 1983 when I was in Max Planck, when I started to work on the PMDA. And it was an assignment, but only to show the pictures of the different types of PMDA, different types of bacterial infections in PMDA. Author was not satisfied. Author always wanted to understand how they being modified and can be utilized for the future world in the form of anti-adherent immune response, AAIR, or in the form of immobilized cancer drugs and whatever it might be. He knows that the most interesting aspects of the nanotechnology is that the surface area. That means due to 10 to the minus 9 meter, the structures so small, its surface area is rigorous. That means all parts of the molecules can be utilized due to nano structure. Now, in that concept, he's moved also with that similar experiment, whatever the PMDA isolation, PMDA collection, PMDA injection, hybrid PMDA injection, their immune response and all these things were studied. Now, these portions which were not studied is the further applications of these PMDA in the form of tube. Now, that in the form of tube means it should be again recapitulated all the process, and also it has to be understood that the donor aspects of the PMDA and hybrid aspects of PMDA, how they could be different. So all these things to be understood, understood in the form of experiment. So without experiment, I believe very strongly the biotechnology processes, biotechnology literature, so whatever in the theoretical aspects will be very minimum. And whenever you do some experiment, then you will understand how vigorous potential activity is possible in the forms of that type of nano level of activities in the microbial world. So microbes are very, very potential in our society, even in the medical science. And nowadays it has been proved the metal extraction, copper, gold, very rare elements, they can be adapted with the microbe. They can be utilized also to isolate that type of low grade metals, especially silver, gold, and the copper, whatever might be coming in our picture. And so we can see in the Gachia as well as in other industrial areas where they extract the copper from the bioleaching in the form of bioleaching, which are being lost by continuous raining process, and so in the form of liquid extraction process in the acidic environment. So these microbes are so smart, they can be adapted also in a very low pH environment to high pH environment. So that is unique, that the many works are remaining untouched or unexperimented to prove that the potential aspects of these microbes. Now, where this thinking process started, origin of the devolution concept of BNT figure C and D. Now, this has been drawn by the author whenever it was observed in the electron microscope. And hour after hours, he was involved in the electron microscope to draw this type of picture. And this has been observed in various types of bacterial isolation as well as the total bacterial system. These are the fimbriae which have been extracted from the bacterial surface antigen, and this is also another aspects of isolation, and that these are the two types of fimbrial structure is isolated in the bacteria, which are the donor and hybrid structures formation. That means these structures of the surface antigen can be changed whenever if it is in the genetically engineered, and if it is another donor system. And so you see that the both are isolated form, but they have two different types of molecular organization. Bio-nanotechnology, nanobiotechnology. Some points I have raised against the merits and demerits. Except side effects, all are merits. That means reduction of size, increasing of catalytic and enzymatic reaction, the major interest of the research and development. The nano business supports the concept of the green and sustainability, the minimization of the spaces and the recyclable natures of the materials. Nano improves the concept of the green and sustainability. Space and time is the global challenge. The developed nations have accepted or have adapted the process and have provided the facility corresponding to energy and the demand of water, health, and food as a common amenities of the everyday life. The developed nations have overruled, controlled the 6 billion populations in the world of the developing world countries. They are increase thereby the maximum facilities, amenities of the enjoyment of the 3.3 billion people of the world which have nothing. That means we must have to start to think how our world will sustain. If 3.5 billion people are not being properly supported to give the facility for the 6 billion people of the world mixed with developed and developing nation, if we avoid the poor nation and the very, very important part of the world, providing they have the capability to provide us some natural products, but we are not very much interested to help them with the concept that without these people we cannot survive. So with that concept, I have to also proceed that beyond that, the nanotechnology is used in naphtha cracking. Nanotechnology is also used in the bioelectric catalytic process and biosensor process, etc. Now you see the carbon nanotube. I've already said, and it was during 1970 or so, that they have always consented to develop single walled, double walled nanotubes, and filtering is one of the best example that was also developed and been studied by Professor C.N. Rao, and who said that we are also very much missing these industries which will be certainly involved in the process of material and medicine, everything processing. So CNT has proved that last 20 years that they have been utilized carbon nanotube in the chemotherapy, but the side effect of these nanotechnology was not possible by that natural chemical reaction and the processing. But we were not aware the last 50 years after pursuit to go with that direction of the microbes, whether they can be also involved in the chemical processes, in the biochemical processes, as well to generate some of the materials from their involvement in the silver, gold, copper components, in the reaction, and to utilize the extracted parts in the process of new materials development, whether it is directly involved or directly applied in the therapeutic system. And BNT will be coming. Why it was more important than the CNT? Because of its double nature. It can develop the immune system, as well as it can deliver the drug to the specific cancer tumor. Why? Why? Because this nanotube has been extracted by genetically engineered donor, means which actually infect the body, as well as develop the plasmid, and that plasmid take the information of that infective nature, can be utilized in the K-12, which has also that type of MDA is being modified after transformation of the plasmid, the informations of the donor into the recipient K-12. So I proceed to say, in case of bio nanotube, this process had the two direction. One direction, it will carry the drugs, means cancer drug. At the same time, whenever they're reaching to that the cancer cell, targeting cell blood during blood transfusion, they can develop also the immune response. Means whenever you are seeing the cancer patient, how a cancer patient dies? A cancer patient dies due to immune support. Means he becomes susceptible against the environment. Means his immune system goes down, and doctors are always injecting some of the infusing drugs, and the side effects without to calculate the side effects. So this is the main reason my wife was killed also in that process, and I have seen how painful it was for a cancer patient if it is actually derives also the side effects. To avoid that, the BNT will be fantastic in future, if possible, to analyze as well as to practically be involved to develop that type of environment. That means immobilization of the cancer drug as well as the immune response of the hybrid BNT isolated from genetically engineered E. coli, that the cancer cell produces the DNA is actually cloned into E. coli K-12. That E. coli K-12 is very smart to accept the cancer cell DNA as well as RNA, whatever it might be, the possible after experimental process to accept. The grows this type of nanotube, also this type of fimbriae, hybrid fimbriae, which can be utilized for the immunization. Now the question is the tube formation, which is still now been not described properly. The tube formation is essential because of the immobilizations of the chemical products, whatever it might be, whether it is chemotherapeutic products or it is only the bioproducts which are being utilized environmentally. Now with that, I proceed this part of the tube formation and their what you can say the transport phenomena, which is a wide range of experimental activities of the chemical engineering. transport process, light food, written book, the transport process, and how the mathematical modeling are involved in the transport process of turbulent and laminates and viscous, Bingham, all these things are coming together. How the molecules can move in a very narrow tubes like a BNT and CNT. How a DASU model, that is one of the physical model which has been improved or imposed or so-called applied in the fluid flow process to measure, and that I've also observed in the bio-nanotube, and how this can be actually applied in the human system as well as in the mammalian, so-called higher groups of animals, in plant cells and everywhere where the tube is nano structure, so not nano. The surface area, the tube surface area, the diameter is a nano level. As you see, our body, arterial system, venous system, and then coming the capillary systems, all surfaces of our body, and which proves that pressure drop can also be controlled by them whenever a patient is very active in the infusion process. Now I move to the process steps developed to prove the BNT from nano channeling clusters. How will you prove it? You have to prove it by experimentally, otherwise nobody will believe it. Then I have proved in that process that you can see there is a syringe, there is a cassette, and there is a syringe, and I've drawn that there are two types of membrane. So all hand drawn, two types of membrane has been involved in the process. My goodness. Oh, chole gelo. Hey, Now we see, these are the picture that I have shown already in the electron microscope. They are isolating fimbriae. You have seen already. We have isolated. Sorry. We have isolated. आगे slide টা যাবো কি করে? কোথায় এটা?
front back.
কোথায় যাচ্ছে না? It is the back. এটা আগে যাব। Sorry. I'm not expert in delivery of no. এরপরে এই জায়গায় I have to prove that this fimbriae has a tube structure, or the clusters of fimbriae can create a nanostructures of tube which support some kinds of drugs, also called some kinds of fluid transport. I have taken a simple polymer araldite. I have mixed araldite with fimbriae in one sense, and other sense I have kept this araldite a pure form. Now see, after observing the not electron microscope, it is in the electron microscope, but this is a compound microscope, high resolutions of 500 increasing size. that you can see the pure aldehyde shows there is no spot. Oh, sorry. Hey, এটা পরেরটা যায় কি করে? That touch করে গেলেই চলে যাচ্ছে। হ্যাঁ হ্যাঁ এখানে
এটা টিপুন পরে।
হ্যাঁ
এই আমরা এটা টিপুন তাহলে হবে।
না না আমি আর টিপব না। ঠিক আছে। So what I see the spotted membrane and clean membrane. That means this aldehyde has no fimbriae mixed, and this aldehyde has the fimbriae of immediate back করার জায়গা থাকবে তো, তাই না?
ওখান থেকে মাউস দিয়ে যাচ্ছেন বলে তো ভুলটা হচ্ছে।
হ্যাঁ এটা কি এটা ব্যাকে যাবার
হুম
আবার কোন আচ্ছা আচ্ছা এইটা না ঠিক আছে মাউস দিয়ে হলে পরে তাড়াতাড়ি হয়ে যাবে। Well, I hope that you understood what I wanted to say. These portions are showing in the form of aldehyde film spot, and this is a single fimbriate band. It's a very thin. It's not properly. A lot of studies are still remains without done, and that's why it will be not very easy to understand at the present condition. But it is a fact that it was worked. The BLT alternative applications, the hydrogen bond application, all these things, theoretically, it is possible. Experimentally, it needs a lot of work. That to be supported by the institute as well as by the government to see whether an electron microscope is inevitable to use, and that to see the way the author presenting all ideas which will be really applicable to the medicine as well as the human system. And I believe it's 100%, I believe 100% that it is applicable and it will be more compatible to the human system in the form of anti-adherent immune response. Yeah, I'm just explaining that things of why. Because before I came that area, I was just innocent in that respect that the extracted hybrid fimbriae can develop immune response. But in further application, I observed in all of the informations gathered in the application, I observed this area has not been studied vigorously by the scientists with the surface antigens or so-called the fimbriae or genetically engineered bacterial stain to develop immune response, at the same time to deliver drugs to the human system, which is a very important at present cancerous world where the patients are actually suffering lots and dying in side effect of the carbon nanotube as well as the drugs application in that process of the CNT. So some literature documents that I have downloaded and placed that how electrochemical processes are being exposed, also called involved in the process of precipitation, in the process of extraction. So here nanobiotechnology and so all these things are coming in the process. Now you may ask yourself as well as to the other, what type of fluid flow will be considered in this type of BNT. It will be considered laminar in the form of, as shown in the figure, laminar types of fluid flow, and in the Dastis model that already been explained. So these are also some kinds of literature's documents where it has shown that nano levels of the drug system is very important at present because of their entry, the hydrophobic natures if it is designed with the world, and how this could be also protected by the other environment of the body. Only drug will be released from their immobilized nature. This is an alternative applications of BNT, that how the bacteria actually colonized on the surface of the human's body as well as intestine and skin. You have seen a lot of skin diseases are also being affected by the microbial process, and that has been also described properly, their settlement as well as their adherence. You see, just logically you say, if a microbes can enter in our body and can infect us, but the process is they may enter in our body easily by means of air, by means of water, by means of food, by means of a lot of other things. But the question is how they actually settled in our body. Is that whenever any bacteria which is enters in our body will respond to some infective natures of the human system? No. Mostly they are not getting any chance to make you infected. Why? Because of their inactive natures, of এই আবার চলে গেল। ব্যাকটা করতে পারছি না, চলে আসছে। এইখানে ঠিক আছে, ঠিক আছে। So you see that if they want to make you infected, then they must have to be adhere in your system, whether it is intestine, whether it is the skin and whatever. If they don't get the chance to adhere, they cannot be infected. So our world is immobilized with various types of microbes. Even the corona is one of the examples that we are not carrying, we are not wearing the mask. Even we say they are not be coming back. But if they come back, they will infect immediately the other because of they found the host. That means host versus graft. That word is an engineering aspects of word which must be capitulated in all the engineers, the host and graft. What is host? The host is we, and the graft, who will graft? The bacteria, corona, virus, or spores, whatever, they come and get inside our body, they must have to graft. That means they must have to colonize. And for that colonizing process, they must have some support. What are the support? The support is the fimbriae. That means there are the very clever from the human system, and they try to overrule our immune system by means of colonization, by means of that type of adherence process. the author already started to think in that way, how I can derive, how I can develop anti-adherent immune response. That means if I made some nanotube, it should be made from the systems where this information of the donor, informations of the infective agent are being carried out. That means either corona or any infective bacteria, that means diarrhea causing bacteria or cough causing bacteria. For example, the corona, which has been killed by the patient, is directly or indirect? No, indirectly. Indirectly means our immune system recognize some pneumococcal tuberculosis type of bacteria always existing in our body or being transformed from the other system. They remain dormant. They are not exposed. And as soon as the corona enters in our body, they started to generate some kinds of immune suppression so that this bacteria, pneumococcus bacteria, pneumococcus tuberculosis type of bacteria is pneumonia type bacteria being exposed. That means they are killing the patient by clogging the respiratory system. That means corona directly clogging is not very well known to us. It is probably in future, it will be directly clogging the respiratory system. Indirectly, it helps pneumococcal type of bacteria, which grows speedily to kill the patient, as well as to infect the patient in that type of microbes. Now, the movement, as I have already said, that this bio-nanotube can be also very, very active. Some gelatinous aspects of the movement, and that has been experimentally proved that some sodium alginate they can develop in the same type of membrane as well as same type of activity. That the same functional thermal management materials, XNA dissipations of the generation capability in the future electronic, they're all being waiting from that type of microbial development grown in the metal water, in copper, silver system, copper, silver, gold, and can be utilized in the electronics process to generate heat and to design membrane type of activity in the process. Macrofluidics. This is a word which is very, very important to understand that in the area of the biology, in the area of a drug transfusion, the macrofluidics plays a great role. How it will be coming out from the immobilized areas to the human system, as well as in the system where they will be active, that the molecule movements in the fluid system, as I have already said, the arterial system and venous system and capillary system of our body similarly acts of the molecular transport in the form of macrofluidics. And for that, some mechanism methods has been studied, how the macrofluidics can be identified, that the pressure drop aspects, and this has already been measured in the form of experiment, some digital pressure drop measurement instrument was attacked in the process of the fluid. In the device that I've already said, the bio-nanotubes were immobilized in the araldite film, and that film was tagged in the form of pressure drop measurement. And I was lucky in the mechanical engineering department of IIT Kharagpur. Some Professor Shumon Chakravarti helped me to measure that the pressure drop aspects of the microfluidics, pressure drop aspects of this membrane. And I have seen really how the pressure drop goes negative whenever it is actually been passed away through membrane. So anyhow, the one thing is that the Dashi model is the most important aspects of our biological and bioengineering aspects of the measurement, where the arterial system and the capillary system can be differentiated on the basis of the pressure drop, and that is also being experimentally proved in the form of that PIMDA to tube formations. So PIMDA and the tube formation. How could you realize it? That PIMDA will create a tube formation? Yes, it can be created. As already I've shown in the electron microscopy, that whenever they are growing, they get or being isolated, they have made in a cluster formation. And whenever it is a cluster formation, you can realize that all PIMDA has some diameter. The ring diameter, whenever they remain in the cluster, they will create some space of the nanotubes, means they will create some space whenever this PIMDA will be tacked with the membrane system. Then they are taking that facility of that nanochannel facility, which has been immobilized with the membrane, has already been proved that the pure araldite has already been proved that pure araldite doesn't show that type of picture in the microscope, and the hybrid araldite, they are giving that type of picture, and proved also that the flow of water. So here, this is a flow of water, proved that the hybrid, wherever the pure, doesn't allow the flow. So that is one of the preliminary development of the PIMDA hybrid membrane with araldite. Simple way to make an experiment and to understand the microfluidic activities in the digital, so-called very sensitive pressure drop measurement system. So a lot of development in the meantime have been developed in the biological responses, which has been characterized in the CFD, computer fluidized dynamics, computerized fluid dynamics, application to biological responses, and mathematical derivations can be interpolated from one type of derived equations to several way to manipulate. unending process of this model of experiment. Only the important thing is to allow a person to do experiment in his way without disappointing and without to do any experiment. No, nothing will come out in that way to express. Please support. There are a lot of things still remaining without understanding. Even the corona vaccines that to be established in future by real knowledge of the adherence, by the real knowledge of the spike proteins of the corona virus, the real knowledge of the air carried corona particles, that how a dry form of corona particles after entering in the body gets so active. So these are the things which I also don't understand. I'm studying. So there are possibilities to fight against the infectious world, but without to do any experiment and to criticize a person, nothing will come out. And don't like to spend the money who will not support to understand this microbial world and their power as already being exposed and proved by our Professor Louis Pasteur. Without Pasteur, it was not possible to survive at the present world without infection. Will be also all perhaps to be killed by any drinks of milk, wine, whatever it might be, because of the microbes, the survivals of the microbes. And probably the rabies infection could also spread. If Professor Louis Pasteur didn't realize the rabies vaccine is also being carried out by the real infected animals. He used a goat or something and dog, whatever it might be, that the anti-adherence concept probably the author has expressed, but the Louis Pasteur at that time probably said our immune response is responsible and important to fight against any vigorous infective world or viruses. So with that, I like to go to the next slides. The lot of development, it's very, very important aspects of the nanotechnology, and especially the surface area adherence aspect has been developed. And finally, I like to say the important bio-nanotube in the future science and engineering is that many areas we are still not exploring. It's a biosensor. Biotechnology is already VSLI, VLSI, very large signaling process, and the software simulating process, and microfluidics, the biological cell interaction, as well as the fluid transport. These are very, very important to design vaccine, to design medicine, to transport medicine, to understand the sustainability of these medicines, and how a bacteria actually can generate that type of environment with the concept of hybrid model of engineering, and the hybrid model of engineering also is require the environment where only experimental environment will sustain support. With that, I like to show these are the reference, which is a very small to read, but anyhow, I published Brahma so last 8 or 9. You can see the bio-nanotube concept was also being carried out since 2009, 2000, 2008, 2005, 7. So with that, I thank you all, the audience, as well as here is the eminent personalities. We are in the webinar's participants to give me a chance to explain, because it will remain in the history the days which that person explained the bio-nanotube, and probably it will be a very, very important aspects of experiments in the field of biotechnology, biomedical science, and bioengineering, whatever it might be you like to say. Thank you very much once again. Thank you. Thank you, Animan, for your
Thank you,
discussion.
Doctor. Thank you, Professor Brahmo. Thank you, Professor Brahmo, for your nice presentation. Now we are having few questions from the members of the audience, and questions from Dr. Sarojit Roy, that can araldite layer be infectious?
Yeah,
Professor,
very good.
Professor Brahmo,
Sure. No, the araldite film cannot be utilized in the human system. There should be some membrane developed where the concept of drug delivery can be implemented. And with that, I like to say this membrane has not been developed till date. It's a sustainability in the body, and it is whether it is infectious or not infectious, I have not gone through. I have only wanted to show you that whether the water, distilled water, whatever, the water is flowing in pure araldite, or water is flowing in hybrid araldite film. That I wanted to show. And as soon as it has proved that the hybrid araldite film provided the response of the water flow, I was satisfied to say that yes, these clusters of film generate nanotube structures because of its mixtures with araldite, and that was also being utilized in the pressure drop measurement. Thank you. Dr. Roy?
So,
yeah, are you satisfied?
So I understand that there is no open question, and so thanking you, Professor Brahmo, for your nice presentation. And now I request our Honorary Secretary of West Bengal State Center, Professor Dr. Raju, to kindly place the vote of thanks.
Yeah, thank you, Engineer Anirban Dotto, for giving me the opportunity to propose the vote of thanks. Is it audible?
Yes. Please carry on.
Thank you. So it's my great pleasure to extend my sincere thanks and gratitude to all the distinguished participants in this technical webinar on the theme BNT, Bio-Nanotube of Escherichia Coli K-12, its engineering and healthcare system. organized by the Chemical Engineering Division of West Bengal State Center, IEI, through this online platform Zoom. So at the outset, I convey my sincere thanks to the panelists, Professor Nitish Kumar Brahmo, FIE, CE, Immediate Past Honorary Secretary, West Bengal State Center, IEI, the Chairman, Chemical Engineering Divisional Subcommittee, West Bengal State Center, IEI. My sincere thanks also goes to Dr. Nirmal Das, Chairman, West Bengal State Center, IEI, and Mr. Anirban Dotto, because Dr. H.O. Thakre, President IEI, is not in this webinar. So still my vote of thanks also goes to him. And Mr. Sandeep Kumar Das, Vice President IEI, and Professor Dr. Shilpa Tripathi, Chairperson Chemical Engineering Division Board, IEI, and Dr. G. Ranganath, Chairman CATE, all are not present in this webinar. Still my vote of thanks also goes to them. So my Mr. Anirban Dotto, Partner and Chief Technology Officer, Messiah Engineers, Joint Honorary Secretary of West Bengal State Center, IEI, and the moderator of this program. So my vote of thanks also goes to all members of the council and division board members of Chemical Engineering Division Board, and all members of the committee of West Bengal State Center, IEI, and all members of the Chemical Engineering Divisional Subcommittee, Director Technical IEI for their and his team for their wholehearted support to make the program a success. Last but not the least, my vote of thanks also goes to those persons who are behind the screen and giving their silent support to make the program grand success. Thank you. Thanking you all. With this, we are concluding today's webinar here. Thank you very much.
Thank you very much.
I request Mr. Sen, Mr. Kinku Sen from IIA headquarters to kindly close the webinar.
লিভ করে যান লিভ করে যান
কি করে যান
লিভ বেরিয়ে যান
Thank you. I'm going out. Shut down. Hey, তুমি কি কফি খাবে?